We've looked at how lymphocytes develop and the various types of lymphocytes. And now we've seen how the antigen-presenting cells, namely macrophages, B cells, and dendritic cells, act to present their antigens to the helper T cells. Now, let's take a look at how the B cell becomes involved in this whole cascades of immunologic events. We'll begin by looking at activation of the B lymphocytes, or clonal expansion and antibody production, just getting the whole thing going. Just as in the T cell activation portion, B cells begin by binding the epitope. So here we're picturing a whole microbe again that's going to be bound by a pre-committed B cell, so it'll only bind the specific type of epitope that its receptor fits. So it's very specific to this microbe. Remember that B cells are also antigen-presenting cells. So just as the dendritic cell did, this cell will take in the microbe and break it down into its components, eventually producing a piece of antigen bound to a class II MHC receptors, and it'll be presented on the surface of the cell. Just as in the previous situation, then the MHC antigen complex is going to be recognized and bound by a T helper cell. This binding happens in a really similar way as the T helper cell did when it bound to the dendritic cell. It involves the CD4 protein as well as the MHC receptor and the T cell receptor. Remember that this T helper cell has previously been activated by an antigen-presenting cell, such as the dendritic cell we saw earlier. These two cells are now engaged in linked recognition, in which the MHC2 receptor bearing the antigen on the B cell binds to the T cell antigen receptor. The T cell is going to now give off additional signals in the form of interleukins and B cell growth factors. This, in conjunction with the linked receptors, serves to activate the B cell. Once the B cell is activated, it signals an increase in cell metabolism, which leads to cell enlargement in preparation for cell division. So we have clonal expansion into these three cell lines. We've got regulatory B cells, memory B cells, and plasma cells. The regulatory B cells are created and act later to minimize the immune response. The memory B cells stop short in development and never become fully differentiated. They will remain as memory cells, floating around in circulation in order to react with the same antigens at a later time and have a much more rapid response. Another group of these cells becomes terminally differentiated plasma cells. These are the ones that secrete antibodies to be released into circulation and go out there and do their job and capture any similar antigens. So in short, during this clonal expansion phase, the stimulated B cell multiplies to produce a large population of genetically identical daughter cells. They'll either become regulatory B cells, memory B cells, kept aside for future invasions, or plasma cells, the ones that are involved in the invasion right here and right now. This figure from your text will be integral in you understanding the B cell response. So again, the process involves the clonal selection and antigen binding, simply picking up the microbe, processing it, and then expressing it on the cell surface. Then we're going to have the B cell and helper T cell cooperation and a recognition phase. This is where the two cells link up using those various proteins on their cell surface. A conjunction of this linking stimulates the release of interleukins and B cell growth factors. Keep in mind the T cell was previously activated by another antigen-presenting cell, perhaps like the dendritic cell that we saw earlier. And the B cell is getting interleukin signals and B cell growth factors. It is now activated. That activated cell grows. More DNA is produced, more cell proteins, more of everything, and then it begins to divide. That's the expansion of the clonal line. So the clonal line expands into 3 different populations: the regulatory B cells, the memory B cells, and then the plasma cells. Of course, the plasma cells then engage in antibody synthesis and release millions and millions of antibodies. These antibodies have the same specificity as the Ig receptor on the original cell, but they circulate in the fluid compartments of the body, where they'll react with the same antigens and microbes shown in the original step of this sequence and continue to eradicate that invader. Let's review this animation on clonal selection to see if we can bring some perspective to the whole situation.
The immune system can produce antibodies against millions of different antigens. B cells, which are constantly produced from lymphoid stem cells, are found in the blood and lymphoid tissues. Each B cell can synthesize only one of the millions of possible antibodies and displays this antibody on its surface. When an antigen meets a B cell having a surface antibody of the proper specificity, it complexes with the antibody. B cells that are not activated do not develop further. Stimulation of only the B cells that carry antibodies that react with antigen is referred to as clonal selection. Following antigen-antibody interaction, antigen-antibody complexes aggregate on the surface of the cell. This is called capping. The antigen-antibody complexes are then internalized. The activated B cell swells and begins to divide rapidly, producing a B cell clone. The B cells then differentiate into plasma cells and memory cells. Plasma cells produce the specific antibody that provoked its formation, whereas memory cells remain in circulation but do not produce antibodies. Memory cells can become activated by a later challenge by the same antigen. Many antibody responses require signals from T helper cells. The T helper cell is stimulated by an interaction with an antigen-presenting cell such as a macrophage. The macrophage ingests the antigen, digests it, and presents it on the cell surface via a class II MHC. The macrophage then activates a T helper cell that carries a T cell receptor capable of recognizing the antigen on the class II MHC of the macrophage. The B cell presents the antigen on its surface using class II MHC. The activated T helper cell interacts with the B cell and secretes chemical signals called interleukins to stimulate the B cell to differentiate into a plasma cell and produce antibodies.
Hopefully the animation helps sort of bring these things together. This material will take some time to organize. One of my best recommendations for you is to pull out a very large piece of paper and diagram all of these components together on one page. Now that we understand the B cell response, let's take a closer look at the antibody structure and function, or immunoglobulin structure. Keep in mind that immunoglobulins are both receptors on B cells as well as the free antibodies that circulate through the body compartments. The basic Ig structure, as we've explored, is 4 polypeptide chains. These 4 polypeptide chains are connected by disulfide bridges, so they have a very strong attachment to each other. Essentially here we're looking at immunoglobulin G. There are 2 arms, and each arm is composed of 2 regions. The antigen binding region, or the fragment for antigen binding, which is this region, and the crystallized fragment, or the F sub C. It was named because it was the first segment to be crystallized for examination. So both segments could be crystallized now, but this one was the first one. And it's the end that binds to the cells or other molecules of the immune system. This figure presents another representation with more of a space-filling model, where basically the antigen binding site is here. This region is highly variable. They call it a hypervariable region because there could be any number of amino acids in there, creating any shape of binding site for a variety of different epitopes. Here's a slightly closer look at how the variety of these binding regions could be. They're super hypervariable. So that is, this region could assume almost any shape. Now, if the antigen binds very strongly into that region, there's a strong activation of that antibody. If the antigen doesn't bind into that site, then there'll be no activation. But it could bind fairly loosely into that site, like it almost fits, in which case we might have a weak activation of that antibody or cell receptor. Now, once these antibodies have bound to their antigens, how do they actually function? So remember, these immunoglobulins can be cell receptors on the B cells, or they could be free-floating antibodies. Here we can see some of the actions of the free-floating antibodies. You have to keep in mind the principal activity of an antibody is to unite with, immobilize, call attention to, or neutralize an antigen for which it was formed. There are a number of different strategies to accomplish this. First of all, the bacterial cell could be tagged with antibodies, so other cells of the immune system recognize it and destroy it. There are special antibodies called opsonins, which stimulate a process called opsonization. This is where the microorganisms or other particles are coated with specific antibodies, so they'll be more readily recognized by macrophages. We can think of this as if they're putting handles on a really slippery object. So alone, these bacteria are quite slippery, but if we put these little handles all over them in the form of antibodies, then macrophages can grip onto them more easily and digest them. Another process is agglutination. In agglutination, the microbes are stuck together by the different antibodies. These antibodies cause them to form a group which renders the microbes immobile and also aids in holding them together in a patch so that they can be phagocytosed by macrophages. They could also be involved in the complement function. So essentially, they work with the complement proteins in immobilizing bacteria and allowing holes to be blown through their surface so the bacterial cell is lysed. Also in neutralization, we might have a virus or another invading pathogen. But essentially in neutralization, the antibodies fill the surface receptors or active sites so that the virus or other pathogen can no longer bind to its target sites. We may also see the production of antitoxins, which is where special antibodies are produced that neutralize the bacterial endotoxins. Or we could see precipitation, where you have small antigen particles that are precipitated out of solution by the binding to a number of different antibody molecules. So as you can see, there are many different strategies for antibodies to attack invading pathogens. Now let's take a look at the variety of different classes of antibodies there are available to do these different jobs. So far, we've seen that immunoglobulin molecules are made up of 4 polypeptide chains interconnected by disulfide bonds. However, there are variations in other molecular constituents of these basic immunoglobulin molecules. Here we can see IgG, which is the one that we've spent most of our time looking at. We also see a number of different other Y-shaped structures, but they could be arranged in dimers or multiple subunits, such as in the pentamer that we see with IgM. We've looked mainly at IgG as a monomer. It's produced by plasma cells in the primary response and in memory cells in responding to a second event. So here's the key information that you need to know about IgGs. We can move on to look at IgAs, and they're a secretory antibody, so they're involved in mucous membrane reactions. They have a couple of neat additional molecules. They have this J chain. This J chain is involved in joining the 2 monomers together to form the dimer form of IgA. Keep in mind that IgA can either come in a dimer or monomer form, so either 1 or 2 subunits. But it's that J chain that joins the monomers in this molecule. You'll also see the same J chain involved in joining monomers in the IgM form of immunoglobulins. The IgA dimer form also has another cool molecule. It's the secretory component. This secretory component is shown here in green, and it's involved in helping IgA move across the mucous membranes. IgM is a pentamer molecule. It has 10 different binding sites on it. This is important because it's produced as a first response to the antigen. So IgM is the initial antibody that's produced. We'll also see IgD in a smaller proportion. This is the one that acts as the receptors on B cells. And finally, IgE, which is an antibody of allergy. We'll spend some more time looking at this in another chapter. Also neat to note is what the percentage of total antibody in the serum is. So IgG is certainly the most popular one. 80% of the antibodies in our serum are going to be of the IgG type. And it makes sense when you look at this whole list of functions that they have. They are our primary antibody. If you had to keep another one in mind, I'd say the next one of importance is going to be IgM, because it's involved in the first response. The key for IgA is its involvement in mucous membranes and antigens that might be found there. So this is a great table to summarize all of the different immunoglobulins. Again, keep in mind that some of them, as in IgD, are receptors on cells. The others are antibodies that are floating around in the fluid compartments of our bodies. Let's have a quick concept check before we move on. What type of antibodies are most important in our mucosal defenses? This one's a quick recall. IgA, IgE, IgG, or IgM. Let's think about some of these quickly. So IgG was the primary one that we've looked at. It's involved in almost everything. IgM was the next most important because it's the first to arrive on any site. So now we're left with IgA or IgE. You'll remember that IgA was the second most prevalent immunoglobulin, and it's the one that's mainly involved in mucosal defenses. And then IgE, if you recall, was involved in allergies and worm infections. So we can learn a lot about how the immune system reacts to an antigen by looking at the antibody levels in serum over time. We consider this number to be the titer, or the concentration of antibodies in the serum. When we consider the primary response versus the secondary response, we're thinking about the first exposure to the antigen versus the second exposure to the antigen. We'll stay over on the left-hand side of the figure here, looking at the primary response. During the first exposure, or the primary response, there'll be a latent period, and then a period where we're seeing synthesis of antibodies. So during the latent period, we lack any synthesis of antibodies. However, it's not as if there's nothing going on in this phase. This is the phase in which all of the things we've been talking about so far are in full force. The antigen is being concentrated in the lymphoid tissue. It's being processed by the correct clones of B lymphocytes. At this point, the plasma cells that are produced from the clonal selection process, where we're producing many different types of B lymphocytes, forming the plasma cells as well as memory cells. These plasma cells will begin to synthesize lots of antibodies, and the serum titer begins to plateau. So in this whole latent period, all the machinery is getting ready for the synthesis of antibodies, and then, boom, we see an increase in the titer of antibodies. This titer will decrease then after a number of weeks or months. We'll notice in this primary response that immunoglobulin M is the first to show up. And as we previously learned, immunoglobulin M is the one that's first responder on the antigen scene, so that makes sense. And then we see a slightly delayed increase in IgG. Now think about what IgG was responsible for. Well, IgG is the one that has the highest titer in our blood, about 80%. It's involved in long-term immunity and memory and production of all of those antibodies that circulate through all the fluid compartments and pick up all sorts of different invading molecules by all the different means we've previously explored. And then our body may not see that antigen for some time. It could be weeks, it could be months, it could be years. But during the second exposure to that same immunogen, the antibody synthesis the titer of antibodies and the length of antibody persistence is rapid and amplified. You see that almost right away, everything kicks into play. IgM, IgG, and the total titer of antibodies in the blood just flies up. Primarily, this response is due to the memory T cells. Because the memory T cells have antigens on their surface, as soon as they recognize that invading antigen again, the whole response is able to come into play super quickly. It's this memory that's the fundamental basis of vaccinations. Essentially, we have an exposure to a dead antigen or part of an antigen. Now we deal more with the epitopes regions of a particular antigen. And so our immune system has already seen it once. It's elicited an immune response and created some memory of that response. So there are plenty of memory cells around. When we have a second response, as in the actual infection, then the immune system can launch a response so quickly that you might not even see any signs or symptoms of that infection.